Hello everyone, I am Vidyashree Somnath. Today I am going to explain about my aim of the project is cloning and expression of human immunoglobulin beta gene in E. coli using pET-28a plus vector. Did you know the single mutation in immunoglobulin beta gene can cause diseases like sickle cell anemia? Today I am going to walk you through how we cloned and expressed this gene, the HBB gene in bacteria and mammalian cell. The HBB gene start codon is ATG and stop codon is TAA. The HBB gene encodes the beta subunit of immunoglobulin, which pairs with alpha subunit to form adult immunoglobulin, the oxygen carrier in our red blood cells. Our goal was to take this human gene, insert it into a bacterial plasmid, express the protein in E. coli, and then conform it in mammalian cells. We used pET-28a plus vector. Why? Because it has a strong T7 promoter for high protein expression in E. coli. It includes a his tag to simplify purification, and it has multiple cloning sites compatibly with the restriction enzymes XHOI and NDEI that we plan to use to get our gene into the vector. We designed the specific forward and reverse primers. The forward primer sequence was CTC, GAG, AGG, AGG, ATG, CAC, GTA, GACT, GAGGA, and the reverse was CGATT, ACGGG, ACC, GGG, TGTTCAT, AGTG, TAA, CAT, ATG. Both primers were designed to have similar melting temperatures around 62 to 63 degrees Celsius for efficient PCR amplification. The PCR step allowed us to make millions of copies of HBB gene ready for cloning. Next, both the amplified gene and pET-28a plus vector were cut with the same enzymes XHOI and NDEI to create matching sticky ends. We then ligated together to form a recombinant plasmid. This plasmid was transformed into competent E. coli cells using the heat shock method, which allows the bacteria to take up the plasmid. We plated the cells, grew colonies, and screened them to confirm which ones had our gene. After confirming the recombinant plasmid, we transferred it into HEK293T mammalian cells using lymphofication, a technique where DNA is packaged with lipid molecules so it can slip through the cell membrane. The mammalian expression step is important because it lets us check how the protein folds and functions in the system closer to immune cells. To verify that our protein was expressed, we performed a Western blot using an antibody specific to the HBB protein. This holds us the size of the protein and confirmed that our cloning and expression worked. In other cases, we could also run enzyme activity assays, drug binding tests, or fluorescence microscopy, depending on the protein's function. So, in just few steps, PCR amplification, digestion, ligation, transformation, and expression, we went from human gene sequence to functional protein expression. This approach not only helps us to study diseases like sickle cell anemia at molecular level, but also opens the doors of therapeutic protein production and genetic research. Thank you.